Okay, guys, we are on one of our last chapters. So this is chapter 21. We have 21, 22, 23, and 24 left, and then we are all done with new material for the year. Then we'll start reviewing for the AP test. So we're going to continue talking about evolution in this chapter, just go over some different mechanisms of evolution. We're going to talk about natural selection again, talk about Darwin's finches again, but let's just start off with natural selection. So just so you know, natural selection, it does act on individuals. And I know, we talked about Lamarck's giraffes and how they tried to stretch their necks up real high, and that didn't work too well just because you can't really stretch your neck and then the neck gets bigger. However, say there is a mutation that comes about. That mutation will affect the phenotype of an individual, especially if it's a dominant mutation. So if we have that mutation present, somehow that phenotype of that individual will be affected. Now, that's only one individual, but if that mutation increases the fitness of that individual, well, that individual will probably do a lot of reproducing, and that mutation will then become more prevalent in the population. And that is how evolution occurs. So that mutation will become more numerous, and then the population evolves. What happens then afterwards is the less fit individuals, or the ones with the adaptations that are not going to increase their survivalship, they're not going to do too well after this. Their traits are probably going to die out. An example of this, let's go back to the finches here of Darwin. You guys can see that this is a very large beaked bird. Okay, same thing here, a little bit smaller, but still, very large. These are going to be the seed-eating finches. Now, when we go to a different area of the world, this is from, a long time ago, 1970s, there was a drought for like a year and a half. There was a drought. So what happened was the food sources changed during that time. At first, the birds were able to still get whatever food they needed. But over time, the food supply changed because the drought didn't give the ability to produce the same foods. So the birds with weak, smaller beaks did not survive because they were used to having the same food source and they were unable to adapt. But the big ones, like the previous ones, all right, these guys here, the ones that were the seed eaters, they were able to eat more than just the seeds. So they were able to change what they were accustomed to and still eat different foods. They were able to survive. The other ones, smaller beaks, they starved. So what happened here, like I said, it was in 1977. After 1977, going on to 1978, they measured the average beak depth, and they found that it actually increased a little bit more. So the beak actually got bigger because the ones with the larger beaks survived and reproduced. So we're going to go over three different parts, guys. We're going to go over natural selection a little bit more in depth. Like I said, we're going to go over genetic drift, and we're going to go over gene flow. So three different things in this chapter. Only natural selection causes adaptive evolution. So when we're talking about adaptive evolution, this is when there are changes in the frequency of certain traits in a population based on the environment or like a change in the environment. For example, let's go back to Jurassic times. We had the extinction of the dinosaurs. And then after that, it gave rise to the mammals. So something changed in the environment for the mammals to be more fit in that environment compared to the Jurassic, the dinosaurs all being there. So something changed in order for mammals to come about. All right. So just to start, we're going to go over section 1. Before it was just like a little introduction. All right. So we're going to talk about genetic variation to start off. So it relates to natural selection a good bit. Right now, the cheetahs actually don't have a whole lot of genetic variation left between cheetah and cheetah, so they're wondering how long the cheetahs are actually going to be alive without going extinct due to that genetic variation. So using that as an example, genetic variation must be present in a population in order for natural selection to occur. If there's no genetic variation, natural selection cannot occur because there are no adaptations or no traits that are more favorable to others. So we're not going to have natural selection there. Genetic variation, also just one more thing about it, guys. It's also caused by the shuffling of alleles in sexual reproduction. So if you remember, we have some alleles that are going to be given to the next generation and some that are not. This does happen by chance in some situations. Remember, crossing over happens where one of them is going to be given on to the next generation and some of them are not. So here is a good picture of genetic variation. You guys can see all the phenotypes there are completely different from one another. We have all different colors of horses there. All right, we can actually measure genetic variation. We just look at the different variables in the genes, different phenotypes. All right. What's nice about this, guys, is that we have homozygous dominant, heterozygous, and homozygous recessive individuals. With that, we can have, at the very least, 2 different alleles. And most of the time, like incomplete dominance and complete dominance, we can actually have more than 2 phenotypes brought about. We might still only have 2 alleles, but we can have more than the just regular complete dominant in phenotypes. So we're going to go back a few chapters, guys, where we went over gene expression. So first bullet point there, genetic variation can be measured at the molecular level of DNA as nucleotide variability. Let's go back to the introns and the exons. So if you remember, the introns were cut out and the exons were left in when we were going through translation to get to our protein. So why do we even have introns then? If we're just cutting them out later on, why are they there? Well, we believe that the introns actually serve as a mechanism for genes that were maybe expressed early in development. All right, so they might have had something to do with us developing, to having our body parts a certain way that they are, that type of thing. Now for the exons, those were the ones that were going to be used to make the protein. And are there variations in exons? Can there be mutations in exons? Yeah, they can. There can be. But sometimes, even though there's variation, we're still going to make the same exact proteins. If we do, no big deal. Sometimes, maybe that amino acid sequence actually changes a little bit, but when it does, and this is going with the last bullet point here, when it does, it's really not a big deal because 99.999% of the time, we are going to catch that mutation in the amino acid before that protein is even used. So it's almost like we're just waiting to actually use that protein for it to take some kind of effect. And when it takes effect, that's when something can go bad with us. But if we catch it before we actually use it, it's no big deal. All right. So even though there are changes in nucleotides, which they can result in like phenotypic variation, we can express them differently. All right. Even though there are changes, most of the time they do not happen. Every now and again, yeah, they will happen. And if everything gets by and everything gets through and we have this protein that has the mutation and it gets through, what this can do is it can introduce new alleles into the population. Those alleles might be favorable. They might not be favorable. If they are favorable, they'll probably stay in. It's a good adaptation. If they are bad, if they are not favorable, if they decrease the likelihood of survival and reproduction, they're probably going to go away and we're not going to see them. Right? So here, once again, guys, we have the exons, the introns. The introns get cut out. All right, you remember that from previously. Okay, so phenotype, we already know that is shown through our genotype. It says also environmental influences. What we mean by this is we mean a genotype that has survived past natural selection. So it didn't get like weeded out. It wasn't a non-favorable trait. Okay, so that's all we mean by that one. All right, we can skip this one. Okay, so new genes and alleles can arise by mutation or gene duplication. We already went through the mutation one. Gene duplication, same thing there, guys. We have a chromosome or a string of DNA. We have duplicated that, so it's still kind of a mutation, and we can actually have new alleles come about because of that. All right, so let's say hypothetically, guys, you have a protein. It has a mutation in it. It's being expressed in your body. Will it go to the next generation? And the answer is sometimes it will. It depends. So it has to be able, that cell has to be able to produce gametes in order for that trait to be passed on to your offspring. If it's a normal cell that's never going to be made into a gamete, we're never going to see that mutation in the next generation. Okay? All right. So point mutations, I know we already went over those. It's just one base pair changes, and we might have a change in our amino acid. We might not. We also might have, if you remember the nonsense one, we might have a premature stop codon, which actually stops the protein from being made. And that's definitely not a good one. So odds are, hopefully, we actually can get rid of that protein and not have it be used. All right? So first thing there, mutations in non-coding regions of DNA are harmless. Yeah, we just talked about the introns, and they probably, a long time ago, helped you guys determine your whole structure of your body. Those ones were used for development. Obviously, you're not really developing that much anymore. You're not like gaining your hands, or I know we talked in the past, like your fingers, how apoptosis occurs in order for your fingers to become non-webbed. If that part of DNA is affected, okay, your fingers are already not webbed. It's not a big deal right now. All right, so these areas, these non-coding regions, they are usually conserved. All right. What happens here, guys, is when we are doing those mutations in the non-coding regions, they are going to be passed on to the next generation. So we might see them or we might not in the next one. But for you, yeah, it's not a big deal. So whenever we have a mutation, guys, that can change the protein, can change the whole amino acid sequence, so it changes the outcome of that protein, and then we express that protein or it's being used, we can alter the phenotype. Now, for ourselves, sometimes it could be pretty much harmless. Sometimes it can actually be bad. All right, if it's like we're expressing a bad phenotype or a completely different phenotype, yeah, that can be bad. But it can also be beneficial if, you know, we are evolving, we are gaining a better adaptation. All right, big one here, guys, but I don't want to talk about too much. So we talked about point mutations there, chromosomal mutations. So these are ones going to be on a little bit of a larger scale. We're going to have a whole segment of DNA, something happening to that segment. So we can have a deletion, we can have a duplication, we can have an inversion, all these different types of chromosomal mutations. They are usually harmful because they are affecting a large amount of DNA. If that DNA that is being affected makes a protein, we're not just affecting 1 or 2 amino acids, we're probably going to affect a lot of them. Okay, so this one, guys, I'm going to talk about from the standpoint of the virus. So mutations in viruses are good. I'm talking about for the virus, not for us, because it keeps any antiviral treatments ineffective from year to year. This is why every year, if you believe in the flu vaccine, you need to get a flu vaccine, because the flu vaccine actually changes, the flu virus changes from year to year. Since the virus is changing, we also have to change the vaccine, because the vaccine might not be effective after a mutation occurred in that flu virus. And it's the same thing that's going on right now with the COVID vaccine. We're finding the new strains in different parts of the world, and they are now testing, the Johnson & Johnson, the Moderna, the Pfizer, to see how effective they are against the new strains. So the virus is already mutating. Again, that's good for the virus. That's bad for us. That's bad for the companies that make the vaccine. But if they make a new vaccine, that's more money in their pockets, I guess. All right. Sexual reproduction. So we talked about recombination in the past. Now, when we have sexual reproduction, this actually can increase our genetic variation in a population because we have a recombination of our genes. So if you remember during crossing over, during prophase 1 of meiosis 1, we have the genes between your 2 chromosomes switching up at different areas. When that happens, when those genes switch up, we are going to get a chromosome that has a little bit of both of your parents being deposited into the sex cell, and then that cell will go and give rise to a kid if sexual reproduction occurs, and fertilization occurs, obviously. So with this, we have completely different genes compared to your parents or their parents. Are they the same genes? Yes, but they are shuffled completely different. So we have the same pool of genes that we can choose from, but we are completely doing it at random. So we're giving rise to a whole new organism. All right. So gene pool, I kind of talked about this in the last slide. This is going to be all the alleles in a population kind of put into one big gigantic area, and these are the genes we have to pull from. Let's pretend we're in Ireland right now, and I know this is stereotypical, but a lot of people in Ireland have red hair. So their gene pool would have a lot of alleles for red hair. Go to Africa. Not a lot of people in Africa have red hair, so their gene pool would be completely different than the Ireland pool, because the Africans would not have a lot of the alleles for red hair, but the Irish would. And that's all. We'll talk about gene pool a little bit later as well. All right. Almost done for today, guys. So we can calculate the frequency of an allele in a population. Some, obviously, are going to have a high frequency, like the red hair in Ireland. Some are going to have a lower frequency, like the red hair in Africa. All we're doing here, guys, is taking our dominant allele and our recessive allele and seeing how prevalent they are in the population. So we got to do the homozygous individuals. We got to do the heterozygous individuals. Now, the homozygous individuals are going to have 2 of the same allele, where the heterozygous ones are only going to have 1. All right, so here are flowers. So this is obviously incomplete dominance. We have red flowers, we have white flowers, and we have pink flowers. Pink flowers would obviously be the heterozygous ones. So let's say we had 320 red flowers. We have 160 pink flowers, and we have 20 white flowers. So here's your first thing to do. we are going to calculate our total number of alleles altogether. So we have 320 for red. All right. So if we have 320 red, we're going to have to times this by 2 because each one has 2 dominant alleles. So 320 times 2, that equals 640. All right. Now, for our, and I'm just going to do all the dominant alleles in blue. For our heterozygous ones, let's do the dominant first, 160. Each one only has 1, so that's 160. So if we wanted to, and I did this down here, if we wanted to calculate how many dominant alleles we have, 640 plus 160, that's 800. So that's how they got that number. Let me do the other ones in red. So we don't have any recessive alleles here for the homozygous dominant. We do have 1 for each of our pink flowers. And we do have 2 for each of our white flowers, but there's only 20 of them. So we're going to add those 2 numbers up, and that's how they got the 200 down there. Okay. Now, if we wanted to calculate the frequency, and they do it on the next page here, but I'm just going to do it here because I think it's a little bit easier on this page. I'm almost done, guys. So I'm not even going to put the lights back on. If we want to do the dominant allele, which if you remember in the past is called P, so our frequency of it would be 800 divided by our total number of alleles. So 800 plus 200 is 1,000. And we get 0.8 from there. So P would equal 0.8. Could also do Q. That is our 200 divided by still our total, which is 1,000. And we would get 0.2 for our Q. Okay? So that's how you calculate the frequency of an allele in a population given our number of individuals of each phenotype. All right, guys. So I think that's enough for a day. Have a good rest of your day, and I'll see you next time.